This video I'm going to show you how to do a basic smear prep for staining bacterial cells. So here I have a clean slide and then I have a wax pencil. What I'm going to do is I'm going to put a nice big circle oval on one half of the slide. Okay, so it's kind of hard to see with the black background, but I have this oval on there. It's my target area. I'm going to turn that upside down so the wax is down. Okay, so now using aseptic technique, I'm going to sterilize my inoculating loop. Follow all the aseptic techniques that I've shown you in the previous videos. I'm going to dip this into my bacterial culture, get a loop full of the bacteria, flame my tube, close that down, and then set my culture down. And then I'm going to rub that bacteria, spread it all around on that slide. I would then flame sterilize my loop, flame sterilize my tube, and I'm going to put another loop full of that bacteria on the slide. Sterilize your loop again before you set that down. Alright, so now I have bacteria on my slide, and we're going to use these slide warmers. Try the back bench here. Bacteria side up. We're going to use that and let that air dry. Alright, so air dry helps them stick to the slide there a little bit. And then we want to heat fix, meaning we want to kill those bacteria. So we normally use the Bunsen burner, but the college has the gas off right now. So I just have a lighter to demonstrate what you would do. If this is your Bunsen burner flame, using a clothespin, not your fingers like I am, what you do is you pass that up and down into the flame of the Bunsen burner 3 times, and that will kill that bacteria. Okay, bacteria side up. So at that point, you're ready to actually do the gram stain. So I'll continue that here and actually show you how to do the gram stain. So here you take the slide and put it on the staining rack here on the sink. Again, it's kind of hard to see. We're going to start with crystal violet. Again, these are chemicals you can find in the bottom drawer. I'm going to flood that slide where I put the bacteria with that crystal violet. I'm going to let that sit there for about a minute. Here we have these DI water bottles. Set the camera up. Sorry, I'm not a professional photographer or videographer. Alright, so let that sit on there for about a minute, and I'm going to rinse that off. Now what's happening is this crystal violet is penetrating all cells, whether they're gram positive or gram negative. So this is our primary stain. If I were to look at this slide with the microscope at this point, all cells, gram positive and gram negative, would appear purple. Okay, at this point it's a simple stain. The next step of this gram stain is to add the mordant, which is grams iodine. So again, you flood that whole area with the iodine. This creates the crystal violet iodine complex. It's basically a large molecule inside the cell that makes it more difficult for that crystal violet to wash out. So let that sit for about a minute, 45 seconds, a minute, and rinse that off. Again, at this point, the mordant doesn't provide any staining, just makes that crystal violet molecule larger. So if I were to look at this slide with the microscope, again, all cells would be purple. The most important step is the alcohol decolorizing step. So what you do is you pick up the slide, get a dropper full of ethanol, if you can. Okay. Hold your slide at a kind of an angle and run that alcohol back and forth. And you'll see these drips eventually go clear with about a dropper and a half of that alcohol. Rinse that slide right away so there's no remaining alcohol, and your decolorizing step is over. This is the most important step. This is one of the reasons why how people can mess this up is if you use too much or too little of that alcohol. At this point, gram-positive cells are still purple. Gram-positive cells have a thick many layers of peptidoglycan, making that cell wall very thick. So that large molecule of the CVI makes it difficult to wash out. You can wash out the crystal violet iodine complex from gram-positive if you use too much of that alcohol. Gram-negatives at this point have a few layers of peptidoglycan, so their cell wall is very thin. So that alcohol is going to destroy that outer membrane that gram-negatives have, and that crystal violet iodine complex is able to wash out of the cell because the peptidoglycan is thin. So those cells are colorless. The last step is we're going to use safranin. This is our counter stain. Flood that slide and let it sit for about 45 seconds to a minute. And what this is doing, it's colorizing those gram-negatives. All right, so those gram-negatives that were colorless are now picking up that safranin, and after you rinse that off, you'll be ready to look at that slide with the microscope. So gram-negatives, they pick up that counter stain. The gram-positives also absorb that safranin, but the purple crystal violet is dominant to the eye, so those gram-positives would still appear purple. All right, this concludes the gram stain and smear prep video.